Bonjour. How you doing? So let's try this again. I already recorded this once, but the video cut out after 2 minutes of a 30-minute presentation. So I get to do it all over again. Doesn't that sound like fun? Anyways, so in previous presentations, have discussed innate immunity, the first and second line of defense, the barrier defenses, and then the cellular defenses and molecular defenses on the inside of the body. And now it's time to talk about adaptive immunity, our third line of defense. What happens if something manages to sneak its way in to our body, gets past the other defenses? Well, then we're going to adapt, overcome, and destroy. This is going to take a while. It's going to take longer to respond than the innate immunity, which is, as far as the skin and mucous membranes, is already there. To develop a strategy to defeat a particular invader is going to take some time, maybe as much as a couple of weeks. It also is going to require contact with the antigen. Remember, antigen is bad, antibody generating substance. And this contact with the antigen, particularly the B cell or B lymphocyte, is then going to cause that lymphocyte to proliferate. That means multiply and form the specialized army of things called plasma cells that are then going to produce antibodies, which are good, by the thousands a second. This whole thing that takes place is called an immune response. And it has two main branches. The cell-mediated involves the T cells or T lymphocytes, and humoral immunity refers to the fluids, body fluids. And whereas this case, the term humor, referring to liquid, is appropriate because these are going to secrete these antibodies that are going to be able to go into almost everywhere in the body. And there's plasma cells producing antibodies. So antigen characteristics and examples. What is an antigen? What are they like? Give us some examples. An antigen is a substance that's going to bind to a component of adaptive immunity. That's either a T cell or a B cell. And these cells have thousands and thousands of receptors on the outside of them to be able to interact with and identify an antigen and find a vulnerability. These antigens are not usually the whole organism, but parts, either a protein or a large polysaccharide that is different from the person's. And these things are the protein capsid of viruses, or the membrane around the outside of them in the case of the coronavirus. The cell walls of bacteria or fungi are certainly not human, so they're going to elicit a response. Toxic molecules produced by bacteria, and even tumor cells of cells that have gone crazy, and they start producing proteins and molecules on their surfaces that are not of that person. So that's how the immune system recognizes these cancerous cells. We can classify them as foreign and self. A foreign antigen, that's mostly what I've been talking about. A structure that's different from a person's, that it recognizes being foreign, that can combine to or with either a T or B lymphocyte. We also have self-antigens. These are like identification badges that tell us, yes, this particular molecule belongs to you. This is you, friendly, do not attack. So that they typically bind with our immune system, but they can. And that's unfortunate. It's called an autoimmune disorder. Auto means self. That's where the body's immune system attacks itself. Type 1 diabetes is an example of that, where the immune system attacks the beta cells in the pancreas that secrete insulin. So let's spend a little bit of time with this very nice diagram here. An antigenic determinant. An antigenic determinant is the actual part of the antigen that we can identify and our immune system can identify and adapt to. It's all based on shape. So what do we have here? We have the antigen. And we have these things that stick out of it. Kind of looks like a coronavirus, doesn't it? And each one of these is an antigenic determinant. This particular antigen has 4 of them. I'm going to use some very sophisticated descriptions. This one right here, I'm going to call bottle, soda bottle. This one right here, I'm going to call notch. This one right here, I'm going to call roundy. And this one right here, I'm going to call pointy. That's pretty sophisticated, isn't it? So what happens is one of these, in the case of a B cell, what happens is it comes over and it's got lots of receptors on its outside, and it feels, literally it comes in contact and actually feels of the antigenic determinant. And once it feels something with its receptors, it says, yes, okay, I recognize that. I can produce antibodies against this antigenic determinant based on shape. So in the case of bottle, the B lymphocyte would identify this shape of this structure and then produce antibodies, which are Y-shaped molecules. We'll be highlighting what they are and how they work more in the next presentation. But on the ends of the Y, the arms of the Y, is a binding site that's shaped complementary to the antigenic determinant. So you can see that right here, and here's a nice fit. This antibody will fit on top of bottle. So once a B lymphocyte, before it encounters, slow down here, before it encounters an antigen, it's called a naive B lymphocyte. Then it encounters an antigen and figures out one antigenic determinant. Then it proliferates, it divides, and most of the offspring cells then turn into plasma cells, which are literally antibody factories, and they'll produce antibodies for almost a week at a staggering rate of thousands a second. Now, this particular antigen has 4 antigenic determinants. So we've got bottle taken care of, but there's others. So a different B lymphocyte is going to come and interact with pointy and make an antibody that has a binding site that's complementary to pointy. And yet a third will deal with roundy, and a fourth will deal with notch. And in this fashion, we can really gang up on this antigen. Immunogenicity, the antigen's ability to cause an immune response. They don't all get the same response. And this is affected by, I'll go over each one of these, degree of foreignness, size, complexity, and quantity. And look at this second bullet and see what it says. Increases in any of these things also increase immunogenicity. So that suggests positive correlation, that an increase in any of those factors increases immunogenicity. So what we know that means then is that the line goes up. So if we put either of any of these factors down here as the x-axis, whatever, and on the y-axis we put immunogenicity, make an I. Let's try for a G. There we go. So, how different is something? If an immunogen is not very different from the molecules of the body, it's not going to create a big, huge. Not going to create a big, huge response. Whereas if it's drastically different, it's then going to create a big response. And this is a fun exercise to go through when we're physically in the class, but I think we can make it work with your imagination. So let us consider that if we were meeting face to face, that for some reason I could get away with requiring every one of my students to wear a white Dallas Cowboys baseball cap. A white baseball cap with a Dallas Cowboys logo on it. I am known to have several of those. And so every day you come to class and you're wearing your white Dallas Cowboys baseball hat, and nobody cares. We don't have a required textbook, so you save money. Hats maybe $15, $20. And that's that. Well, let's say somebody comes, they want to infiltrate our classroom. And they know that they have to wear a white Dallas Cowboys baseball cap, but they don't have one. But they have a white hat with the Houston Astros logo on it, which is somewhat star-shaped. And they come walking into our classroom, and we not notice because it's not that much different. We'd have to take a real close look to notice that that logo was different than the one everybody else is wearing. What if somebody came in wearing they knew that they got the word that they had to wear a cowboy hat, but instead of the white Dallas Cowboys baseball hat, they come in wearing an actual cowboy hat, like they just come from the rodeo. Well, then that's going to stand out as being much different than anything anybody else is wearing, isn't it? For sure. And so that's much different. That degree of foreignness is high. What if, heaven forbid, they came into our classroom wearing a green Philadelphia Eagles hat? I would think that we would have to jump that person and give them a beatdown because, Philadelphia Eagles. All right, so that hopefully represents an example serves to highlight the degree of foreignness. Size is the same thing. If something is really small, like a viral particle, it might go unnoticed. Whereas something is larger, it's going to be more noticeable. All right. If you want to talk about something uncomfortable, nobody talks about the golf ball in the room. What do we talk about it being in the room? The elephant in the room. It's hard to ignore an elephant in a room, right? Why? Well, because it's so darn big. So larger molecules are going to elicit a bigger, quicker response than a small one. Complexity. That in the previous example, this one had 4 antigenic determinants. That's going to cause a big response. What if instead of all those little points sticking out of there was just one, and it was small? It wouldn't elicit as big a response. The more complex something is, the easier it's going to be recognized that it's foreign. And quantity. If it's just one, maybe you don't even notice. But if it's a whole bunch, you're certainly going to take notice of that. So any of these 4 things, as they get larger, are going to elicit a larger response. And then there's a sad case of autoimmune disorders. That's where the immune system doesn't tolerate a very specific self-antigen. And what do they do? So the body attacks itself. Rheumatic heart disease is an example. Type 1 diabetes, I've mentioned. Multiple sclerosis, the immune system attacks the myelin sheath of neurons in the central nervous system. It's not good at all. These people might be on medications to suppress their immune system. And that's going to make them more vulnerable to infections because their body's not going to be able to tell the difference between something that needs to be attacked and something it's being told not to attack, which also then is going to place those people at higher risk for things like the COVID virus, which is why we'll hear about that in the news. The T and B lymphocytes, they each have unique receptor complexes, like I said, thousands, maybe 100,000 per cell. These are like keys that they use to try and various locks, tools to identify these antigenic determinants. The B lymphocyte is going to make direct contact with the antigen. They've got to actually feel of it. And more on, that seems out of place, more on that in just a minute. Organ transplants and MHC molecules. What is MHC? MHC stands for Major Histocompatibility Complex, and it's the main identification badge for a person's cells. It indicates that, yes, this is you. It's like literally like an identification badge that has your name, your picture, and some sort of, I don't know, number on it to identify you. Your driver's license. So when somebody's going to receive a transplant, we want the MHC molecules, a major histocompatibility complex, to match up as good as possible. We also have to match the blood types. The ABO blood group has to be compatible, and the RH has to be compatible as well. But the problem is nobody has the same exact MHC molecules except perhaps identical twins. So they'll do a blood test and try and figure out, how different is the donor and the recipient, and they'll try to match that up as best they can. But then the immune system is not going to know. There's no way to really tell the immune system, hey, immune system, you see that new kidney in there? I kind of need that new kidney. Would you leave it alone? You can't talk to the immune system like that. It's going to try and reject that organ. And so that person is going to be on high levels of immunosuppressive drugs for the rest of their life. And so that's another person who's at risk for infections like the COVID virus. Now, I know you're spending a lot of time. You're very busy with this class. I told you in the first day it was going to be crazy, and I think now that you realize that I wasn't kidding, it really is. But when you're going to need some relaxation, 4th of July weekend and onward, there's a book that deals with the MHC molecule called Robin Cook. Oh, it's written. It's not called it. It's written by an author named Robin, like Batman, and Robin Cook, like milk. No, I'm a chef. Robin Cook is also a physician, so he writes thrillers that have involved medical science, and his book is called Chromosome 6, because in humans, the MHC molecule is located on chromosome 6. And in his book, what they do is, now this has to be a rich person. Rich people, not me, pay to have their major histocompatibility gene spliced into an embryo of a bonobo. And you might say, a bonobo? What's that? Very few people know what a bonobo is. There's a bonobo. Look at the face. Hey, buddy. Anyway, so a bonobo is a primate that's just a little bit smaller in body size than a human. And in the book, and this is not entirely out of the realm of possibility, the book was written about 15 ago. They spliced you can pay to have your MHC gene spliced into a bonobo. Now, of course, this is both immoral and illegal in most places. So this part of it, the story takes place on an island, right? We got to have it on an island like Jurassic Park so that they can't escape. An island off the coast of Equatorial Guinea. Does anybody know where Equatorial Guinea is? Mary Lou, do you know where Equatorial Guinea is? Most people don't know that either. It's a sub-Saharan country on the west coast of Africa. And so here's this island with these bonobos with human genes spliced into them. What could possibly go wrong? I don't want to give you a spoiler alert, but when you start messing around and transplanting human genes into a close relative like the bonobo, what could possibly happen? What could possibly go wrong? Could the bonobo decide they want to have something to say about that? Robin Cook, Chromosome 6. You can get it on audiobook. You can get it in an app. I listened to it one time when I was on a long road trip, and fascinating book. All right, the B lymphocyte, before its first stimulation, is naive, and upon its first stimulation, the antigen binds to a receptor molecule, BCR, and it's no longer naive, and then this causes the B lymphocyte then to proliferate, We know what that is. And then differentiate, that means change into, most of them differentiate into plasma cells. And they're the ones that turn into the virus factory. Some of them, though, become memory lymphocytes. We'll talk about those in a minute. They're going to keep their BCRs. That just means B cell receptor. So, but the BCRs, these B cell receptors, are the ones, they keep the ones that identified the antigen. And they have long lifespans. They can weeks, days, months, sometimes even our entire rest of our lives. These B memory lymphocytes can live. There's a little diagram here. So here's the receptor is actually found to have fit the antigen, and then the B cell comes over to a T cell, helper T cell, and says, "Hey, look what I found! I can do this." And the T cell activates the B cell to go on to produce plasma cells, mostly plasma cells that produce the antibodies against that antigen. One type of antibody, not the four like in the early example. If for an antigen with 4 antigenic determinants, it's going to take 4 B lymphocytes to make those 4 different antibodies. And then some will have some memory cells, and the memory cells, they'll retain those B cell receptors, BCRs. So these plasma cells, they form antibodies. The effectors of humoral immunity. So the B cells, the plasma cells, migrate their way into the lymph nodes where they hang out and proliferate and produce literally gazillions of antibodies. The effectors of humoral immunity. So where is that going to go? They go to the lymph nodes. They're producing their antibodies in the lymph nodes, and then lymph, of course, is going to transport those antibodies and return to the circulation near the heart. And the way they'll be in the circulatory system, and they're eligible to go pretty much anywhere. They can come out of the circulatory. They're small enough. They can come out of the circulatory system in the capillary beds, flooding the interstitial fluid and bathing the cells, going back into the lymph and circulating around almost everywhere in the body. A couple of exceptions: the cerebrospinal fluid in the central nervous system. The antibodies can't go there. They have a hard time getting into the humors of the eyes and the capsules of synovial joints. But other than that, they can go pretty much anywhere. They produce millions and millions and millions of antibodies during their approximately 5-day lifespan, and they go everywhere in the body, and eventually they're going to find the bad guy, the antigen, and they're going to bind with it, and then one of 3 things might happen. We'll talk about that in the next presentation. A measure of the level of circulating antibody is called the antibody titer. That's just kind of an antiquated term, but that's just a measure of how much antibody is in the blood. So I was talking with somebody last night that a friend of mine that had suffered with the COVID virus back in March, and he's an older fella. He's in his late 60s, and he said he got sick. He didn't need to be hospitalized. He didn't have any breathing problems. He said he had some serious muscle cramps for a couple of days, and it was miserable. And he's okay now. It's been I guess a month and a half, and he said they measured his antibody titer, and he said that he was like off the scale. It's a normal is 0 to 100, and he said his was off the scale, like way over 100. So I thought about kidnapping him and stealing his antibodies. That way I could give them to my daughter. She'll survive. Yes, it's my daughter's the one that's COVID positive. She's okay. She's asymptomatic right now, but it is worrisome. All right, so we need to talk about antibodies. So let's do that next. Till next time, au revoir.